We have already talked about DNA repair when we were talking about DNA replication. Now we are going to talk about some other proteins that are responsible for maintaining integrity of DNA. We are going to talk about P53 protein. It's also called guardian of genome. It plays a very important role in controlling cell division. So let's look at that. P53 protein has many anti-cancer mechanisms. It can activate DNA repair proteins when DNA has sustained damage. P53 can also arrest the cell cycle at G1S boundary regulation point. If DNA damage has been recognized, it will hold the cell at this point, G1S transition, long enough for other cellular machinery to go in and repair the DNA. If DNA is repaired, P53 will release the brake and cell will go into cell division. However, if cell is unable to repair its DNA, P53 will guide cell towards apoptosis or cell destruction. Of course, it can initiate apoptosis, the programmed cell death, if the DNA damage proves irreparable. P53 is also one of the most commonly mutated genes in human cancers. Of course, if it is the guardian of genome, it is involved in many processes, including arrest of cell division and guiding cell towards apoptosis. If there's a problem, if there's a mutation in this gene and its product is non-functional, the results will be catastrophic. And of course, many types of cancers have a mutation in P53. P53 can sense the DNA damage through accessory proteins, one of them being ATM. It is a protein that can sense DNA damage. P53 also has an anti-proliferative effects and also regulates apoptosis. The normally unstable P53 protein is stabilized by DNA damage, so its concentration increases. So if the cell is working perfectly normally, cell doesn't need P53, so P53 is made and it is degraded. However, if the cell detects there is a DNA damage, it will not degrade P53 and allow P53 to do its various functions. One of the functions of P53 is it works as transcription factor. P53 induces expression of P21. I hope you remember, I just told you that P21 is also a very important protein that can inhibit cell cycle. We saw that in one of our previous modules. It can inhibit almost all CDKs. I mentioned it when we were talking about cell cycle. So how does this work? So when there's DNA damage, the P53 is stabilized. It senses the proteins that sense DNA damage. They allow P53 to survive and not be destroyed. P53 basically acts as a transcription factor that increases the transcription of P21. P21 can bind many cyclins and their CDKs and result in arrest of the cell cycle. So how does this miraculous P53 work? So let's assume a hypothetical situation that there has been a DNA damage due to X-rays, for example. There are protein kinases that can sense DNA damage and they will add phosphate group to P53. P53 will become phosphorylated and now P53 will be able to survive. Unphosphorylated P53 is sent to proteosomes for degradation by a protein called MDM2. When cell is working normally, MDM2 will bind P53 and send it to proteosomes for degradation. However, if the DNA has been damaged, there are protein kinases that become activated, sensing the DNA damage, and they will phosphorylate P53, and now P53, phosphorylated P53 will not be able to bind MDM2 and it will survive and it will not be destroyed by proteosomes. P53 then binds to regulatory region of P21 gene. We have talked about that, and results in production of P21 gene, transcription, translation. P21 will now act as an inhibitor protein. It will bind the CDK and cyclin complex and it will make it inactive. So here we have seen the role of p53 as transcription factor. We'll also look at the role of p53 as a protein that can regulate apoptosis also later on. So phosphorylation of MDM2 or decrease in MDM2 production can also cause increase in p53, which will allow the production of the p21 gene. So here's a broader picture. DNA gets damaged by X-rays. It results in activation of kinases which phosphorylate p53. p53 can no longer bind MDM2 and be degraded. p53 becomes acts as a transcription factor, binds regulatory regions on DNA adjacent to the p21 gene, which results in production of p21 gene product, which is p21, which binds and which inactivates the CDK and cyclin complexes. Another way p53 plays a crucial role is that excessive MYC production. I hope you remember the MYC gene we talked about earlier. Abnormally high levels of MYC cause activation of a protein called p19ARF, which binds and inhibits MDM2. So previously we saw that p53 survives MDM2 by virtue of phosphorylation by kinases. In this case, abnormally high levels of MYC can result in production of p19ARF, which inhibit MDM function, causes an increase in level of p53, which can also cause either cell cycle to arrest or cause cell to go through apoptosis, depending upon the type of the cell and extracellular environment also. That plays a role in how this p53 is going to function. I've told you that p53 is a very important gene and mutation in it can cause cancers. Here I will give you an example of one of these diseases. Li-Fraumeni syndrome, for example. One of the p53 alleles is mutated, which causes a 25-fold increase, 25 times the chance of developing a malignant tumor by age 50. So when there's a DNA, we have already talked about it, so I'm not going to spend too much time on it. If there's DNA damage, p53 level will go up. Either the cells will be arrested in their particular cell cycle, the cellular machinery will get a chance to repair the DNA. If repair fails, cells will go through apoptosis. However, if the p53 is not there, the cell cycle will not be arrested, cells will not go through apoptosis and will result in a malignant tumor. So here is an example of disease that occurs if p53 is mutated. Also, viruses have developed a strategy. I've told you many viruses can inactivate p53 and also RB protein. For example, papilloma virus uses two proteins called E6 and E7. They sequester the host cell's p53 and RB proteins respectively. Also, SV40 knocks out these proteins. However, it uses a dual purpose protein which can actually knock out both these proteins, the RB and p53. The single protein can neutralize these two proteins, which will be in the benefit of virus, because viruses proliferate in the cells, and if there are more cells which are infected by these viruses, it will be in the interest of this virus to knock out these proteins. So we have seen genes that actually repair DNA, what happens if they are mutated, and how that can result in cancer.